Have a nice and safe day to all my viewers. I again wish you a very warm welcome for this channel. Dear students, in the last video we have already discussed about amino acids, peptides, proteins and two different structure of protein. We have already discussed about primary structure and secondary structure of protein. So, the rest part of protein conformation, that is the tertiary and quaternary structure, will be discussed in this video. Okay, let's discuss about the tertiary structure. Up to secondary structure, it has been found that only structural proteins are produced up to secondary structures. Okay, suppose this is the alpha helix, this is the alpha helix, or in case of beta pleated sheet we can only describe this structure. These proteins are not able to represent any biological functions. Biological functions cannot be achieved by such proteins. So, in these cases, for achieving, these are the structural proteins for different biological function. Biological function, it need to be folded more further. It needs folding further. So, the biological functions can only be achieved when secondary structure again folded. Clear? So, there is a need of folding in secondary structures. The folding of secondary structure represent this folding of for biological function. The folding produces tertiary structure. Okay, tertiary structure. The tertiary structure again became complex to produce quaternary structures. In this way, the complexity of protein goes on rising. Okay, suppose we will take the example of secondary proteins. Suppose I am taking the example of fibroin, the example of keratin. These are fibroin, keratin, these are secondary structures. This can be only represented by a structural part. For achieving biological functions, these secondary structures are not sufficient to perform the biological functions. So let's discuss the secondary and a tertiary and quaternary structures. Detail about tertiary structures. Okay, as we know, suppose the biological function of a protein can be achieved by foldings. Suppose this is a alpha helix. The alpha helix means the helical arrangement of protein. It is secondary structure. The biological function is only possible when alpha helix coiled further. How? Suppose this is the alpha helix. Again it became coiled. Okay, so this structure is further coiled, alpha helix further coiled to represent tertiary structure. This coiling is stabilized by bonds, different type of bonds. What are these bonds? This may be hydrogen bond, it may be hydrophobic bond, it may be ionic bond, it may be disulfide bond, it may be van der Waals force. Okay, these are the different type of forces which help in aggregating the secondary into a more complex tertiary structures. We'll discuss about all these linkages. What is hydrogen bond first? First come to hydrogen bond. This bond, this hydrogen bond, it is formed by formed for the purpose of further folding. This is hydrogen bond. What is this bond? This bond is the tendency of hydrogen atom to share electron with two neighboring atoms. Tendency of hydrogen atom to share electron with two neighboring atoms in the chain. So this is called as hydrogen bond. Tendency of hydrogen atom to share electron, okay, with neighboring atoms. Clear? Then hydrophobic bond. What is hydrophobic? This hydrophobic bond, this bond is formed between the non-polar side chain, okay, the non-polar side chain of neutral amino acid. Non-polar side chain of neutral amino acid have a attraction. This attraction is hydrophobic bond. Then comes to another bond, ionic bond. Ionic bond. We know the ionic bond is otherwise called as salt. It is the attraction or bond which is formed between acidic and basic part of amino acid. This is formed by acidic and basic part of amino acids. Okay. So after ionic bond, there is disulfide bond. Disulfide. What is this bond? This disulfide bond is the linking between two cysteine amino acids, cysteine and cysteine. These are the between two non-adjacent cysteine residue, the disulfide bond is formed. Then the ultimate attraction is van der Waals force. Van der Waals force. It also helps in stabilizing the torsion. It is the electrostatic attraction between uncharged molecules. So it is electrostatic attraction of uncharged groups. Okay, so these are the different type of forces which help in stabilizing tertiary structure. This tertiary structure is represented by suppose we will take the example of myoglobin, the oxygen carrying molecule in muscles. The myoglobin represent this tertiary structure. Then come to the last structure of protein, that is quaternary structure. What do you mean by quaternary? What is quaternary structure actually? It is the last complex structure. Okay. After that, there is no further complexity in the folding. What is happening here? Suppose this is the tertiary structure. Folding of tertiary structure produces this folding of secondary produces tertiary. When two or more tertiary again coiled, suppose this is one tertiary, another tertiary here, another tertiary protein, there is another tertiary here, tertiary structures. Okay, suppose another tertiary structure is here. I am taking the example of the hemoglobin, tertiary. Four tertiary structures, they are again linked. These are linked by different type of bonds: hydrogen bond, disulfide bond, ionic, hydrophobic. These interactions exist between them. There are two alpha gens and two beta gens. Okay, so two alpha, two beta gen, alpha gens and beta gens. These are joined producing quaternary structure. So this quaternary, which is represented by hemoglobin here, is the folding of tertiary structure. So the tertiary structure is more complex. There is not any further complexity in the folding after tertiary. So it is all about the structure and conformation of protein. In the coming video, we will be discuss about the other biomolecule, that is lipids, carbohydrate and so on. Thank you all.